So meiosis can be really confusing unless we have a very strong understanding of chromosomes. So I really want to make sure we reviewed this first. Here we have a homologous pair of chromosomes. What makes this homologous? A lot of students get confused between that and like what is sister chromatids. So this is homologous. And what it means is that you have dad's present and mom's chromosome present. And we're going to get into the idea of diploid and haploid and all that going forward. But first, what I wanted to also make sure that we have down is gene versus allele. These here are genes: hair color, body height, something that you need in order to exist, like traits that you need in order to exist. So you have to have a color to your hair, right? Or you have to have a certain height. You know, you need an eye color. You need like skin color. You need all these things. And genes are describing those general characteristics. Whereas alleles are going to tell you whether your hair is red, whether that gene is going to code for you to be tall. That is what alleles really are. They're like specific instructions on what the gene will express. From a mathematical standpoint, it's like saying that hair color, like genes are functions and alleles are inputs to those functions, right? Genes are general equations, whereas alleles are the specific values that go into that equation. So hopefully that helps some of you all out. So to be haploid, what that means is that you have a cell that maybe only has dad's information in it, right? Only dad's genes. This is haploid right here. So like, oh, okay, so you have red hair and, you know, you're tall. And even if you have two of dad's chromosomes in there, right, of that same chromosome, right? So this one's still going to say tall, red hair, and this one here is still going to say tall. This is still considered haploid because it's only dad's information. It's only half of what you need in order to survive. Every cell needs both mom and dad's information in order to properly function. So let's get a good idea on chromatids versus homologous pair. Once again, this is homologous. And what I'm going to show you here is what happens after S phase during the cell cycle. If this is dad's and this is mom's, well, dad's chromosome is also going to copy itself during S phase. And then mom is also going to have her chromosome copy. So there's still only two chromosomes here. Even though I just, drew, it looks like you want to say 4 because you see 4 things here. But you have to remember that this one right here that's outlined in red, which is dad's sister chromatid now, is just that, a chromatid. It's a copy. So it's not considered a new chromosome. It's just considered a copy of a chromosome. And then you have mom's here, which is outlined in black. That's her chromatid, her sister chromatid. So even though we see 4 things here, there's only 2 chromosomes. But what you can say there are 4 of is there are 4 chromatids. You see, I'm trying to spell and talk at the same time. Chromatids. There are 4 chromatids, but only 2 chromosomes, because there's really only 2 sets of information, dad's and then mom's. Okay, so once again, if you're haploid, what that means is that this is an example of a haploid cell, only dad's information. And then if I circle only mom's information, that's another example of haploid. Even though there's 2 chromosomes, or chromatids, there's only 1 chromosome in that green circle here. All right, so let's get into the song of dance that is meiosis or meiosis. So the names are the same as mitosis, right? Where you see prophase, metaphase, anaphase, telophase, and cytokinesis. But there's 2 rounds of division, 2 sets of division. And you see it here where there's prophase 2, metaphase 2, anaphase 2, and then cytokinesis and telophase 2. And really the only difference between these 2 is like what chromosomes are present. But the general movement is the same. Whereas in prophase, you're going to see the chromosomes condense, right? Here, you're going to see them still organize. And then here in metaphase, they get into the middle. Metaphase 2, they get in the middle. Anaphase, they separate. Anaphase, they separate, so on and so on. But what you really want to keep track of is what chromosomes are present in these different types of forms and functions and all these different division types. So first off, prophase, we already talked about it, but I want to get over into is a tetrad. A tetrad is this image right here. When you see that dad's sister chromatid and mom's sister chromatid are present. That's when you have 4 chromatids, but it's still considered 2 chromosomes. This here is a tetrad. And in prophase, they end up getting joined, all right, in a place that's called the charismata, and that's when they actually have their crossing over. Remember, crossing over is when they exchange bits of the chromosome in order to ensure that there's a lot of genetic variation and variability. People look different and their genes are somewhat different. So that's when tetrads form, is here. And metaphase, you see them all line up there in the middle again. So that's the same as mitosis. And then anaphase happens. And what's really important is the anaphase is we see that the tetrads are split. But that kind of looks, that's obvious in the picture, but what's more important is that the chromosomes are split or separated. And why is that so important? Because if we look back here to this, it's not that one dad and one mom are going into each new cell. It's dad, dad. It's dad, dad, and then mom, mom. They're both chromatids from this are going here. Let me make this very as clear as possible. And then both of dad's chromatids can go in here. So what you end up with, and this is a question that comes up on a lot of exams, is that at this point, the cells are considered haploid. All right. If we look at our original count here in prophase, we see that there are 2, 4, 6 chromosomes. 2, 4, 6 chromosomes, yep. Right, but then there's 2, 4, 6, 8, 10, 12 chromatids. 4 times 3 is 12. Yeah, chromatids. All right, and if you look here, there are 1 chromosome, 2 chromosomes, 3 chromosomes. There's only 3 chromosomes, but there's, let me write that down, but there's 6 chromatid. Right? So that's what makes it haploid. Not that this number has been cut in half. That's actually not that's not what's causing it to be haploid. It's the fact that the chromosomes have now been cut in half. In this bottom cell here, only dad's information, let's say dad is red and mom is blue. Dad's chromosome is the only one that's present here. Whereas mom's, which if they were together would make it a homologous pair, mom's version of that same chromosome is now above. So that means that this is haploid, that is haploid. And that's an exam question that comes up very often. And that is after which phase do we consider the cell to be haploid? It's at telophase or cytokinesis, or at the beginning of prophase 2. Honestly, these things are so hard to differentiate based on what teacher preferences are. So it's really clear that you speak to your teacher. But I promise you, by this prophase 2, this cell here is haploid, N, and this is haploid, N. Whereas this cell here is 2N, which is diploid. And then in metaphase 2, you see them separate again. Line up in the middle, then separate. And then here in telophase and cytokinesis 2, they go and once again separate into 4 completely different cells. And these are considered gametes. Now remember, gametes are what the whole purpose of meiosis is which I should have started off with, but it's when you're making either sperm or eggs. That's when you do meiosis or meiosis. It's when you're making sperm or eggs. Anything else is going to be done through mitosis. So you get 4 gametes, so that's 4 sperm. Or if you're a female, you make one egg, and then you make really one egg. Let's pretend that's the egg that you make that a female would produce, and the other three are kind of like used for their spare parts to ensure that this egg has more than enough to survive for a long time, whether it's in the uterus or in the womb or when it's being fertilized. So either way, you make 4 gamete cells, whether you're male or female. But if you're a male, you have 4 viable sperm, but females only make 1 viable egg. Best of luck on your exam.